We have a very packed program, which is always exciting. So welcome. For those who were not here last night, welcome. For those who were, welcome again. This is our Beyond Pesticides 34th National Pesticide Forum. We started at the year we were founded, which was 1981. That's 35 years ago. And I guess people ask me, well, why isn't it the 35th? And I guess we missed a year in there somewhere. But here we are. This is an important event in the cycle of our work at Beyond Pesticides because we really feel that it's important to get people together, it's important to network, it's important to share cutting-edge information. But the really important element of what we do and what you ultimately do in your communities is to understand the science that drives the action. So we've spent a fair amount of time during this program today talking about science and our understanding of what the interrelationships are between the exposure of pesticides and impacts on human health and the environment more broadly. Our goal is not to just talk, obviously. Your goal is not to just talk, but our goal is to institutionalize change so that the decisions of governmental bodies reflect not only the science, but our values and our principles at the community level. And those values and principles go to stopping toxic assault on human health and the environment. Last night we heard from Rachel Carson, as portrayed by Kailani Lee. And Ms. Carson, as a marine biologist, science writer, a poet, author, she warned us to respect nature and support it, harness the benefits it offers us, but never think that we can overpower it. The real-world examples, when you actually dig into Silent Spring, are eerily similar to those issues we deal with today. Impacts on salmon, aquatic organisms. These are things she wrote about: birds, beneficial insects, human effects. She warned us that pesticide use is related to rangeland, utility, brush control, forestry, agriculture, private lawns, parks, and golf courses were all issues that were back then, and that's many years ago in the 60s, of tremendous concern, and they continue to be of concern today. Many of us organizing have found some resonance on the crisis that we're seeing with pollinators today in this country. And in our quest to find the next best chemical solution to problems, the chemical industry came up with neonicotinoids. And as you know, these are systemic chemicals. And, you know, it's interesting because as pest control has evolved over time, we've talked about integrated pest management, now we talk about organic management. But all of these management systems are systems, in effect. They are systems of monitoring, they're systems of evaluating the conducive conditions that give rise to a pest problem. And yet, here we are in the 21st century, and we've introduced a chemical, a systemic chemical that's introduced or into the plant either through the seed, right, because we're talking about seeds that are coated with a chemical, neonicotinoids, and then they move through the vascular system of the plant and they express themselves through the pollen and the guttation drops and the nectar. And along come insects that are feeding and foraging and pollinating. And of course they are, along with the target insects, all the insects that are necessary for a balanced ecological system are being indiscriminately poisoned. Now, what could be further from an organic system or even an integrated pest management system than the prophylactic use of a chemical that indiscriminately poisons? So certainly one could argue that the reason those chemicals were introduced, or this family of chemicals, was to reduce human exposure in the environment. So for our quest to reduce human exposure and eliminate human exposure, we've introduced the concept of indiscriminate ecological poisoning. And that's because we have not yet been successful in making this transition to alternatives and alternative ways of thinking. Now, when we do our daily work, and when you go before city or county council and talk to elected officials about this issue, the first thing you talk to them about is the science. And often what you find is disagreements over the science. Many of the disagreements are on EPA risk assessments and the findings of acceptable risk. We talk about the limitations of what EPA knows in terms of vulnerable population groups, people with pre-existing medical conditions, interrelationships related to mixtures of chemicals and synergistic effects. These are unknown issues, but we also talk about what we do know, and we're going to hear from some of these scientists today about what we do know. We know a heck of a lot. We know a heck of a lot, but we also know very little at the same time in terms of the complexities of the impacts of these chemicals on biological systems. And so one of the elements that is missing from our public policy is this concept of precaution. When we don't know as much as we think we should know, and I think if you brought a group of scientists together, they would tell you we don't know as much as we should know, then the question is, should we move ahead with this escalating chemical dependency when in fact we have huge gaps in information? So that brings in the concept of precaution, and you may hear today throughout the forum some people talking about precaution as a public policy issue and as it relates to pesticide use and pest or management. So our goal in the course of this work is to work with nature, not against it. And last night, the amazing talk by Kristen Olson related to her book, The Soil Will Save Us, I think captures the fact, and I mentioned this last night, that there's a complexity that we really don't understand in the soil, which for me really reflects the broader complexity of the environment generally. And because of that complexity, we basically have ignored key elements of that coexistence that we should be evolving with as human beings, evolving with nature. It's interesting, one of the books that we refer folks to is a book called Teaming with Microbes, and we heard microbe farming last night as another term for regenerative agriculture or soil health agriculture. But here's a review of Teaming with Microbes, a book I recommend to everybody that came out in the Seattle Times a couple years ago. Said, smart gardeners know that soil is anything but an inert substance. Healthy soil is teeming with life. Not just earthworms and insects, but a staggering multitude of bacteria, fungi, and other microorganisms. When we use chemical fertilizers, we injure the microbial life that sustains substances. But there is an alternative to this vicious cycle. We can garden in a way that strengthens the soil food web, which Chip Osborne will talk about later today. The complex world of soil-dwelling organisms whose interactions create a maturing environment for plants. So we convene this conference at a time when the solution is within our reach. And I, like Kristen, see this situation with a lot of optimism. I see opportunity in the lack of knowledge, because as people become aware of this situation, they move to action, or they are moved to action. And Maine, of course, is a tremendous model for advancing action, so we'll be hearing from a lot of the folks today who have grappled with these very issues and adopted ordinances. We'll be hearing from a council member, former president of the Montgomery County Council in Montgomery County, Maryland, who played a tremendous leadership role in advancing new ways of thinking about how we manage land within a political subdivision, a political jurisdiction. Our job, of course, is to work with leaders to help them be influential in changing the thinking on this very issue. At the same time, we also work in the marketplace, and this is another exciting piece that you all are working on as well. And we have at this conference folks who are grappling with this at the point of purchase, where people are making either good decisions or bad decisions from our perspective in terms of health and the environment. And clearly in a marketplace where people are used to buying a product with a 4-step program, applying a chemical and seeing results, that's a huge shift, that's a huge change, and it's a cultural change that has to take place. So we'll be hearing from people that are there face-to-face with consumers who are making these decisions as well. Now, I recently finished a 5-year term on the National Organic Standards Board, and that, for me, was an opportunity to delve pretty deeply into what is allowable in organic systems, or what is allowable in organic production. And Kolau Bandera from Hawaii is in the audience. He just finished his position on the NOSB last year. Jim Gerritsen, who will be speaking later tonight, attends all the NOSB meetings, National Organic Standards Board, as a farmer. The Maine Organic Farmers and Gardeners, MAVCA Association, participates heavily in this process. But not a lot of you are participating in that process. And I want to make a plea for us getting much more engaged in what may at times appear to be a bureaucratic process that is far off in Washington somewhere, controlled by bureaucrats and people that are not in tune with our values, all of which is true, and our principles. We have a tremendous law in the Organic Foods Production Act, a law that was actually put together by farmers and environmentalists and processors and certifiers. And when that bill was put together, we didn't see organic potato chips. We didn't have snack foods that were organic of all kinds. And so obviously as craft foods gets into the organic food market, some of us see that as a good thing. We're expanding the demand for organic commodities. But we're also, in many ways, people have a dissonance, I think, when they see a product and they look at it and they say, this can't be organic. How can this be organic? It's counterintuitive. What that comes down to is the work of the National Organic Standards Board, because a material doesn't make its way, or a product, to market unless there's a particular material that it depends on. And evaluating that material for its impact on human health and the environment, we question whether that product is needed, that ingredient is needed in the product. These are all issues that affect you as a consumer in the marketplace. The bottom line here is we want to see the organic market grow, but we want to see it grow without this disconnect, or in connection and relationship to our values and our principles. And believe it or not, the Organic Foods Production Act, I believe, embodies our values and principles. To the extent that we as a community, as individuals, as parents, as community members, as consumers, do not engage that process and keep that process accountable to those values and principles, we will see a watering down of those standards. But at this point in our history, we have an incredible opportunity, and the opportunity is organic. Organic provides us with a holistic and cross-cutting solution that integrates concerns about clean air, water, safe food, and as we'll hear Will Allen and Kate Duesterberg talk about tonight, protection against global climate change, sequestering of carbon. This is happening at all levels of government, and you all are the critical piece to change. Beyond Pesticides is committed to working with local communities across the country. We are so proud and honored to work with Protect South Portland and Portland Protectors in advancing changes in policy. The mayor of Portland will be here later today and hopefully endorse that effort as well. But these, when you see a unanimous vote of the council, the city council of South Portland, endorsing the idea of organic land management within the city jurisdiction, those are victories that the people of South Portland are proud of, and Protect South Portland should be very proud of. So I thank you, Rachel, and Portland, for all the work that you've done to bring us to this moment. And this is being replicated across the country. So let's roll up our sleeves, delve into the science, as hard as that is sometimes. Think about how we can use that science and make sure that the scientists throughout this conference are making this information accessible to you so you can convey it to elected officials, policymakers, decision makers. Because we are on the cusp of some amazing changes. We can harness all the energy in this room and throughout the country. Have the models. Maine starts with an M for a reason in my mind, and that is because it is a model in so many ways. And the work that's been going on here, either by ballot initiative or city council votes, is leading the nation, and with MAVCA for 45 years, has led the nation on change. So congratulations to you all. Thanks for being here. Enjoy the organic food throughout the conference and use it for nourishment so that you can stick with this program till about 10:30 tonight, because I'm telling you, the speakers at the end of this conference are the bedrock, along with Heather, who is the bedrock in Maine here, for moving change, the change we want to see in our communities in Maine and across the country. So thank you so much. I'm going to turn the program now over to Routt Reigart, who is going to introduce the plenary speakers. Thank you. Is Routt still around? Okay. Oh, Routt, okay. Get a picture of this, please. So Routt, let me just tell you a little bit about Routt. I don't want to take up a lot of time, but I first met Routt. I had seen Routt at different meetings over the years, but I first got up close and personal with Routt when he chaired the EPA's Children's Health Advisory Board. And Routt was sitting there in this room next to the EPA administrator and the director of the office and saying all these things that I couldn't believe I was hearing at EPA about how we need to protect children and children's vulnerability. Routt is a pediatrician, has served on the faculty of the Medical University of South Carolina for many years, has done clinical work throughout those years, and in his spare time, I don't know how, he has managed to be up in DC, chairing committees, getting extremely involved in the advocacy side of science. So he is a tribute to all those scientists and medical practitioners that really understand the connection between what they're seeing in the human population and the need for policy change. So Routt, thank you so much for being our board president and for being a bee and a pollinator at the same time.
And thanks to David Wheeler, who made me into a bee. They said I wouldn't do it, but I did. It's so out of character. I'm viewed as a relatively straight person. But for those of you that weren't here last night, he's just introduced me. I'm Routt Reigart. I'm excited to see all of you here. I'm excited to be back in Maine. I love Maine. And thank you all for being here. But my job this morning is to introduce Aaron Blair, who's going to speak to us as the first keynote speaker. He's chair of the International Agency for Research on Cancer, which everyone else calls IARC. It takes a minute to figure that out. Working group, not the whole thing, but working group. A branch of the World Health Organization announced its finding that one of the world's most popular pesticides, glyphosate or Roundup, is a likely human carcinogen. He ran the National Cancer Institute's Occupational Studies branch and is the author of over 450 publications on occupational and environmental causes of cancer. And I have to say, I've read many of his publications, but didn't have the opportunity to meet Aaron until this morning, and I'm glad you're here. He's received the National Institutes of Health Directors Award, the Public Health Service Special Recognition Award, NIH Merit Award, the DHHS Quality of Work Life Award, so on and so forth. He's won them all. And interesting, near the end, the NIH Director's Award for the Deepwater Horizon Gulf Oil Spill Study, and he's served on many review groups. But I told Aaron this morning that I wouldn't take his whole speech up by introducing him, but it's hard to cut it short. So I'd like you all to welcome Dr. Aaron Blair.
It's a pleasure to be here this morning to meet old friends and make some new ones. Hopefully, what I'm going to do today is sort of go through a series of slides and discussions about how we carry out research on pesticides. Many of you probably already know this, and I'll put a spin of sort of the researcher-scientist view on it, which means it has some possibility of being completely deadly, because that's what we're really good at. But give it a try anyhow. Hopefully that it will be of some use to you as you take action in trying to use information to make policy decisions and move policy decisions. I just mentioned I was on the IARC working group that looked at several different pesticides. IARC is now beginning to do that. There's enough information to begin a more further evaluation for cancer than has typically been available in the past. Should say that through my presentation I'll be making suggestions of things that we ought to do, looking at additional research. And since I started this and doing this in 1976, a fair reply from you might be, well, you've had your chance, you know, you haven't accomplished it now. Let's get somebody in there who obviously knows what they're doing, but what I've done is what I've done. So today the overview is just a little bit about the research process, how we evaluate human studies in particular, and then finally something on research needs. Pesticides are a really unique group of chemicals, as you know, that from my point of view, looking at it from an occupational and environmental epidemiologist, we know a lot of different chemicals cause human harm. Benzene causes leukemia. Arsenic causes bladder cancer and lung cancer. I can go through this long list, many of them that you know also. What's unique about them is they were not designed to harm people. They inadvertently harm people as they were used. Pesticides were designed to harm living systems. So that by itself says we ought to be a little more worried about these suckers than a bunch of other things, because we know it's going to do something to some living organism. Maybe a plant, and maybe they're enough different from us that we don't have to worry, but I think that's actually not the case. So designed to do harm, we know they actually accomplish their pesticidal action through a lot of different mechanisms. Not just one, a lot of different ones, depending upon the pest, of course. And because of this, they have opportunity of having have effects on a lot of different diseases and symptoms. And of course, we know they're really widely known, or widely used. So here's just a list of different outcomes that have been linked to pesticidal use. the amount of information that makes this linkage, how strong it is, varies. For some of it, it's pretty strong, some of it, it's pretty weak, but these are just different things in studies that have shown up both in animals and in humans. The list is long, and it grows every single year. It gets larger. The one I find it's interesting here is actually obesity. Now, who would think pesticidal exposure would have anything to do with the fact that too many Americans look like me? You know, and it has things to do with how our body processes energy, how it stores it, and that sort of thing. And it's not just pesticides, it may be other things too. But who'd have thought this would be one of them? So mechanisms by which pesticides might operate are actually, there's a considerable number. They're ones you can think of right off the bat. Maybe you know these anyhow. Some pesticides cause gene mutations, some of them cause chromosomal damage. A recent group of pesticides we find actually interfere with the length of the telomeres at the end of the chromosomes. If you don't know what these are, telomeres are like a little plastic thing on the end of your shoelace. They keep your shoelace from unraveling. Telomeres keep the chromosomes from unraveling. They can get too long, which means that genes don't function well, and get too short, which means it's unraveling. A whole bunch of pesticides, not a whole bunch, several pesticides do this. Pesticides cause oxidative stress. It just means it creates chemicals in cells that they themselves are dangerous. It's not the pesticide now, it's the chemicals. They're free radicals, they do a lot of bad stuff. Pesticides interfere with the immune system, with endocrine systems, obviously impairs nerve conductions. That's how they kill insects and different sorts of things, has thyroid disruption, and some pesticides appear to speed up cell replication. You think, well, that's not bad. Well, but it is bad, because the more times the cell divides, the greater the chance is it will make a mistake. And the more replication turnovers you have, the greater chance of mistakes, and some of those mistakes will be bad, and clearly works for cancer. So IARC, in the beginning of 2015, really had hardly looked at pesticides. The list on the left, which is this on your side, on the right, on the left on your side, just shows what they had made pronouncements about at the beginning of 2015. NTP had done slightly more. But since beginning of 2015, IARC has looked at a list of other pesticides, and for every one of them, they classed them in some category, suggesting we ought to worry about them for cancer. IARC just deals with cancer. And you can see the list here. And it's already been mentioned that glyphosate was one of those that was ranked as a probable human carcinogen. If you're not familiar with the IARC ranking, that word itself tells you pretty much what it means. Possibly isn't as strong of evidence as probably, and the upper category, of course, is there is sufficient evidence, and we call it a human carcinogen. It's either things like benzene, smoking, HIV, wide variety of different things. So a little bit about glyphosate. Again, you probably all know this, but it's a really widely used herbicide. It's getting more widely used all the time, used with genetic modified crops. There have been other reviews. The European Commission reviewed it in 2002, said no problem, essentially. German Institute for Risk Assessment, which makes early evaluations, it's then turned over to the EU, 2013, said no problem. IARC did it in 2015. In early 2015, and since then, the Risk Assessment, Institute of Risk Assessment, have reevaluated and evaluated the IARC evaluation. They said, well, IARC had it all wrong, and there really isn't any problem. So there's this battle that goes on, and it happens in these sort of evaluations, sort of the first time in. You deal with the first group that says we ought to worry about this factor, or whatever it is, there's almost always a pretty big backlash. No, that's actually understandable. The backlash comes from 2 points of view. Not all scientists agree. Different groups of scientists can take the same pile of data from different studies and look at it and come to slightly different conclusions. This is how science operates. People debate, and eventually things get settled. This debate about glyphosate, whether or not it does anything or not, that you see in the news in different places now, it went on for benzene, it went on for arsenic, it went on for dioxin, it went on for tobacco for 50 years. So this is not unusual. This is what happens. So you have both the scientific debate, and then you have economic debates, the issues that enter into it also. So here is the evidence that led IARC to conclude that glyphosate was probably a human carcinogen. from rodent bioassays, which IARC looks at rodent data, bioassay data, human data, epidemiologic data, and mechanistic data, and as well as what exposure information there is. From the rodent bioassays, the rodents got renal cell tubular carcinomas, hemangiosarcomas, and pancreatic cell adenomas, and skin cancer. Now, you may not know what those are, but the key thing is these are rare cancers. I mean, they're really rare. So if you see something that's excessive a little bit for a tumor that occurs all the time in rodents, well, you're comparing it with those that weren't exposed. It's a fair comparison, but it doesn't raise your hackles the way one does where you get really rare tumors. These are really rare. So that makes it stand out. These just don't occur typically in most studies. Mechanistic studies said that glyphosate caused DNA damage and chromosomal damage and oxidative stress. Every one of those we know is related to cancer. And epidemiologic studies found an association with non-Hodgkin's lymphoma in the US, in Canada, and in Sweden. But as most epidemiology, if you looked at this, it's never all that clear-cut. And one study, the agricultural health study that I was involved in, it's a huge cohort study in 2 states in the US, in Iowa and North Carolina, didn't see an excess with glyphosate. This is the way human data usually are. You have to look at the totality of what's going on. The IARC evaluation process, I want to just spend a little bit of time on it. It's composed of working groups of 14 to 30 people, depending on how many things you're evaluating. The world's literature is reviewed, and the literature is animal studies, human studies, mechanistic studies, and exposure studies. This working group drafts documents before the meeting. So there are already drafts available to look at. Then you come together at a meeting, and it's run from a week to 10 days, depending on how many things you're doing, and you go over the drafts, you discuss the papers to try to get ready to make a determination of which category you ought to fit these substances in. The categories are, it's not likely to be a human carcinogen. There's data, but we really can't tell. It's just too noisy to make any judgment whatsoever. Possibly a human carcinogen, probably a human carcinogen is a human carcinogen. And then mechanistic data are used to modify these circumstances. And just a little bit about how different studies get into these various categories. For sufficient evidence, if you have sufficient epidemiologic data that the scientists conclude this stuff causes cancer, that makes it a category 1. Doesn't make any difference what happens in animals. Doesn't make any difference what happens in mechanisms. If the human data are sufficient, it's sufficient. For probable, you can sort of get there in different ways, but usually it is limited evidence in human, means there's some studies that show something, some that don't, just like glyphosate, and there's sufficient evidence in animals, and that puts you in a category of probably. And you just don't put it in sufficient because the stupid human studies are messy. Don't tell you exactly what's going on. I do human studies, by the way. I recognize the mess. Possibly it says there's probably limited evidence in humans and insufficient evidence in animals, or sort of the flip of that. Inadequate evidence in humans and sufficient evidence in animals. Either one of those 2 scenarios will give you in this category, a probable category. Mechanistic data can increase your evaluation or decrease it depending on what the mechanistic data says. So for example, from the human, if you had an evaluation where there was limited evidence in humans, sufficient evidence in animals, and the mechanistic data was overwhelmingly positive, you might bump it to sufficient. On the other hand, if you had limited evidence in humans and sufficient evidence in animals, and the mechanistic study said that the way that the animals get the tumors are through a mechanistic process that cannot occur in humans. There are things like that. Animals are different than we are. It cannot occur in humans, you might bump it down. So the mechanistic thing is sort of an arbitrator that moves things up and down. That didn't happen in glyphosate, it stayed where it was. So I wanted to spend just a little time about human studies, what goes on. First thing is to figure out, who you're going to study and what exposures are. And there's a lot of way to get different exposures and to look at different types of studies. You can have just monitoring, look at ecologic patterns, case control studies based on self-reports, death certificates, or you can do cohort studies. So first thing is how is the pesticide used helps you design the study, what factors affect intensity, and what protective practices might be employed to do that. So here's just getting an idea of where it's used. This is atrazine. You know, it's used in corn and soybeans and different corn and different sorts of crops in the U.S., and this bright red area is sort of where it's really used. So if you really wanted to study occupational exposure to atrazine, that'd be a good place to go. Go do your studies there rather than some other parts of the U.S. where it isn't used so heavily. How things get applied make a difference in what the level of exposure is. This is what happens in orchards. Orchards, the trees are up in the air, and you have to spray the stuff up in the air, and you can see this fog of stuff in that picture going up in the air, which means it's going to drift around, including on the people who are applying it. Field crops might be a little different that aren't orchards. There you're typically spraying down. That doesn't mean that it doesn't volatilize and go up with wind currents and so forth, but the initial technique is a little bit different. Different ways to do it in fields. You can have it purely mechanized. Here you got individuals hanging on stuff, spraying as they go along. Animals are a different deal because you have to be around the animal usually, which means that can bring you in close contact with them, and you touch them, they touch you, you get exposures. And of course, individuals do things either with little bitty gadgets or bigger gadgets. Just want to show you a couple scientific slides that show how difficult it is to figure out what exposures are. So this is looking at two different chemicals, atrazine and 2,4-D. These are measurements that we made in the urine of farmers. And we did these measurements over a whole year. So we went at baseline, which is essentially in the winter, and followed over the planting season, growing season, back to the winter and the off season. The green are the farmers' levels. What is color is that? Purple, blue, something, is non-farmers from the same county. So people resided in the same area. So you see what happens. For the non-farmers, you know, maybe they have a little bit something, but not very much. Stays the same throughout the year, and that's true over here for 2,4-D also. But for atrazine, of course, for the farmers and the non-farmers, levels are about the same in the winter. Farmers start doing things, levels go up. You can't use things like this and not get it into you. I mean, it's literally impossible. How careful you are depends on how much you get into you. So during the growing season, it goes up, comes back down to the fall and the winter of the next year, drops farmers' levels, drop back down to the non-farmers' levels. So that's atrazine, a pattern you might anticipate. Look at 2,4-D. So here's 2,4-D, here's the non-farmers in the winter. The non-farmers and the farmers are different. Now, where are farmers getting 2,4-D exposure in the winter? I guarantee you they're not spraying weeds, but it's somewhere in their environment. When they start using it, it goes up, comes back down to the next winter, still up. So this stuff is around in their environment. It's in their pickup, it's on their tractors, it's in their house, I don't know where it is, but they're getting exposed, and the non-farmers aren't, which points out the difficulty of getting exposure right. Things are sometimes just not the way you anticipate. Here's another slide that sort of shows that. So here's a case that this is boom spraying. Again, farmers doing it. So here's a case that individuals didn't have a cab, didn't have gloves. Now they go in, we look at people who have a cab. You would think a cab ought to protect you. You know, you're in a tractor cab, and many tractor cabs now are air conditioned. They weren't when I grew up on a farm, but they are now. You think that'll keep it off of you? No, you get more exposure. So you do something you think ought to work, it gets worse. You look at gloves. Gloves really make a difference. You put gloves on, boy, it drops the level down. This is where you get exposure, mostly if you're using this stuff. You get it on your hands and in other parts of your body, and it goes through the skin. The inhalation part is usually small. Of course, the flip side of that would be if you kept it entirely off your skin, all of your exposure would be through inhalation or food or something. So this just shows the difficulty of figuring things out. Cabs didn't help, made it worse. So studies that we do, I want to just run through the types of studies a little bit, because sometimes there is an impression that there are certain designs that don't provide useful information, they're too noisy. That's not true. Literally all studies are useful. They're useful to a different degree. Some studies have more problems than others, but it usually means almost always, they provide some information. So I'm just going to start with sort of the simplest sort of thing you do. One of the easiest things you can do is just look at the pattern of a disease geographically. These are called geographic studies or ecologic studies. And the problem is you're not studying individuals per se, you're studying groups of people. These are a series of maps that the National Cancer Institute did in the middle 1970s, plots mortality rates for cancers by county in the United States. So this is total cancer. And what you see, of course, is it's really heavy in the east, particularly in the cities. Well, why would total cancer be high in the cities? Because city people, are decadent, not like farmers where I grew up, and they smoked. And that drives total cancer rates. If you look at lung cancer, it'll look exactly the same. So this is lung cancer. This is the pattern you get. Now you look at leukemia. It looks nothing like that. Bright red areas out here in the center part of the United States, from north to south. And even if you look in the individual states, this is Omaha. It's the biggest city in Nebraska. It's not elevated. Other places are elevated. So whatever's going on for high rates of leukemia mortality is not due to the urban lifestyle. It must be due to the rural lifestyle. Doesn't mean it's pesticides. They're around engine exhaust, they're around dust, they're around a lot of different things, but it's the rural lifestyle, which gives you clues. So these studies are useful to help direct where you want to go. As a matter of fact, we did this. After this studies came out, the NCI published these maps. You could do that. Next step, you can take information and try to go the next step. An easy way to do that is with death certificates. So you get people who died of a certain cancer, get people who died of something else, you look at what occupation they had. And it turns out for leukemia, non-Hodgkin's lymphoma, prostate cancer, if you do that in these states where center part of the United States, farmers have higher rates than non-farmers. Well, again, suggests, okay, something to do with agriculture, you should look at that. Now, you can go even further, you can characterize the county by the type of agriculture that it has. And it turns out for leukemia, living in a corn farming county is a little greater risk than living in some other types of counties, which again helps focus a little bit on what the details might be. The next step is to actually get direct information from individuals. And that direct information comes from interviewing or you get from records and so forth. These are interview studies from farmers and non-farmers. This happened to be in Kansas looking at non-Hodgkin's lymphoma. And you find a very nice exposure response trend as the more, the more days per year that they work, they use these particular herbicides. In Kansas, it's almost all 2,4-D. The risk goes up. So it's a six-fold excess for people who use it more than 20 days per year. So this is very precise in terms of what the chemical is and what the exposures were. Other things enter in. Farmers, growing up on a farm, I can say that back in my day, you change clothes when your clothes were soiled. And spraying pesticides does not soil them the way mucking out a hog barn does. So you didn't necessarily change at the end of the day, and certainly didn't change when you finished applying the pesticides. And if you leave it on your clothes, it's in contact with your body, you have a greater time for getting exposure, so that's where this question came from. When did you change your clothes? The longer you wait, the greater the risk again for non-Hodgkin's lymphoma and herbicide 2,4-D, actually. The other thing we need to, so these are looking at individual pesticides, and I think we have to go that route for almost everything. It's just my sense to move society, move regulation, but that's too simplistic. That most people don't get exposed to just single pesticides. In fact, as humans, we don't get exposed to a single anything. We're a messy species. We got stuff all over that we get exposed to, and it's true for pesticides. So one of the things you might think, well, let's just see if additional pesticides, however many you get exposed to, makes a difference. So here's a study where we just counted. People told us what they used. We added them up. Some people used more than 5, some 2 to 4. No difference whatsoever in the risk. None. Relative risks are all 1. But if you looked at it just a little bit differently, suppose you said, we're not going to, for cancer, we're not doing this for non-Hodgkin's lymphoma for any pesticide. We're only going to count the pesticides for which there is some evidence they might cause cancer. And when you do that, you get this pattern, which shows an exposure response gradient. Now, why would that make a difference? You think about throwing together all sorts of chemicals that do nothing towards the disease that you're interested in, then of course it just waters down those that do. So if you accept the belief that not all pesticides cause every cancer, I believe the evidence is overwhelming, that is true. It says when you lump them all together and deal with them as an entire group, you're likely to miss stuff. So you have to get on the track to look at the ones that are important. That's why you have to go to individual ones, and from that then build back up to looking at combinations, because combinations may have additional effects. So I wanted to say a little bit about a large study that I've been engaged in since the early 1980s. It's known as the Agriculture Health Study. It's essentially all the farmers in Iowa and North Carolina and about 2/3 of their spouses, and we enrolled them in the early 1990s, and we're following them on through today, where we gathered information on about 90,000 individuals. We interviewed them multiple times. We did it when we enrolled them. We did it 5 years later, 5 years after that, and just recently finished enrollment. So now we can look at a lot of different outcomes in relationship to a lot of different agricultural and lifestyle issues for people on the farm, of which pesticides drove this effort to carry out this sort of study. It's known as a prospective cohort. They take a long time to really get somewhere. They cost a lot of money. It's why you don't see them so much. So here's just comparing the cancer rates in farmers in this cohort with the general population in those two states. Well, what's interesting about it is, first of all, for cancer overall, farmers don't get as much cancer as other people. You know, it's 15% lower for the farmers and a little bit more for the spouses. But it's not true for every cancer. Some cancers, there are excesses, excess of ovarian cancer among women who were applicators in this study, slight excess, but a real one probably, of prostate cancer, a possible excess of multiple myeloma. So farmers are interesting in it, not just for cancer. Their mortality experience as a whole is down compared to the general population. Whatever these people are doing, they're doing a lot of things that are right. That they don't smoke as much, they don't drink as much. Lack of physical activity is typically not a problem that farmers have. You know, so there's three good things right away that makes you healthier. Doesn't mean they don't have other things that aren't a problem. And so we're trying to look at these other problems. And this just shows the complexity of cancer. Non-Hodgkin's lymphoma overall is elevated in farmers in many places and linked to some pesticides, but you sort of look at this little trend across levels of use of non-Hodgkin's lymphoma. Well, I don't know, if you really were a believer, you might say that's going up, but it's not much. And every confidence interval is below 1, so that it's not statistically significant. But if you look at subcategories of non-Hodgkin's lymphoma, every cancer has these histologic subcategories, even though we talk about them if they're single one thing. That non-Hodgkin's lymphoma is the worst of all, but you just think about what the name means. We're not saying it's Hodgkin's disease. We're saying it's a lymphoma, but it's not Hodgkin's disease. Well, that must mean it's a lot of things, and that is true. It's a lot of things, and this suggests that these different things might have different risk from individual exposures. This is for diazinon. Looks like the chronic lymphocytic leukemia and small cell lymphocytic lymphoma might. A follicular cancer, a follicular lymphoma look like it might. So it's the precision of the details of figuring out what's going on. Diazinon and NHL also. Diazinon and lung cancer. Lung cancer has the different types. We're most familiar with squamous cell. Smoking really does it, but it also does adenocarcinoma. They appear to have slightly different causes, at least at different levels. So here we see it looks like there might be something going on with adeno, not so much with the others. Here's just some other sites related to diazinon, ovarian cancer in women, but not breast. Maybe say a few things about organochlorines. Chemicals have been around a long time. Most of them aren't widely used anymore. We all still carry them in our body, of course. But I don't know, 90% of us have DDT or its metabolites in our fat cells. So here again, breaking down non-Hodgkin's lymphoma for different subtypes, and it looks like there might be difference compared to NHL overall. And atrazine, I'm just sort of running through a bunch of these so you can see there's really differences by things. So here's atrazine might be related to ovarian cancer in women who use it, but not breast cancer. Well, you know, often they go together, but they don't always go together. Thyroid cancer also, statistically significant. And here's again a case talking about multiple exposures. This is just comparing two things, atrazine and acetochlor, and that dual exposure bumps it up and makes it statistically significant. Just using those two, where by themselves they don't show much. I'm going to skip this. Multiple myeloma is an interesting one because we know a precursor thing. It's monoclonal gammopathy of undetermined significance. And you can look at this. We did in the agricultural health study and found that in our cohort, they had about a twofold excess. These farmers had about a twofold excess of this. Literally all multiple myeloma is preceded by this condition. And it was twice as much as what was found in Olmsted County, where Mayo Clinic is, where they had looked at this a lot. So it suggests that farmers might have an excess, and you can look at particular groups of cancers and different exposures. And this is a case where we look at permethrin, where it looks like there's a trend for it. So I've spent a lot of time on cancer because that's what I do. But I think everything I talk about in the complexity of doing it implies to all the other outcomes also. The difficulty with the other outcomes is sometimes that outcome is not as easy to determine, so it makes it even harder to do the studies. But here's just the links that we found out of the agricultural health study for different pesticides with different outcomes. So 2,4-D, I talked about it in relationship to cancer, but we have associations with wheeze, with asthma, with telomere shortening, and with depression. You can read the list there and see the variety of different outcomes that have been linked in the agricultural health study. That doesn't mean proven, it just means linked in the agricultural health study, which says we ought to worry about them. So what do we need in the future? I think it's very clear, we need studies that look at a lot of outcomes. There's no way to go at this in a simplistic manner, just looking at a few things. There's tons of things that should be looked at. Some of them may prove not to be linked truly to certain pesticides. That would be true for sure, and maybe even for any pesticides. We need to look at studies that look at combinations of exposure. Nobody has just one. We all have multiple ones. Even if we're bystanders in the general public, we still have more than one exposure. We need studies that looked at susceptible groups. I'm sure we're here today about issues with children, but there's other susceptible groups. The elderly, rapidly moving into that group myself, maybe already there. People who are immunocompromised, people who have other diseases, all those things are important. And we need mechanistic studies that tell us how the substance might be operating in humans. Be happy to answer any questions you might have. Yeah, back here.
Oh. So I'm just wondering if you're aware of any studies that have been done on, the lawn care pesticides, because, it is applied at a much greater rate, and you have that constant re-exposure that people, re-entry exposure that, say, like somebody who would live in a property that has been, sprayed or pelletized, 4 to 6 times a year.
You're asking about not the people who are treating the lawns, but the people who have their lawns treated.
Well, kind of a both and, because I know that they do some monitoring of people who work in the industry, but I think it's pretty minimal. There have been studies certainly of people who apply the pesticides from the business end of things, usually,
typically.
Healthy young white
Right.
males versus the people and the families who live in this stuff.
There's some suggestion that, for cancer at least, that there might be a linkage there. There have been studies of people who apply pesticides on golf courses, and there were sort of the farmer cancers that occurred there: non-Hodgkin's lymphoma, prostate cancer, leukemia. I don't quite remember about leukemia. So that's the case. Actually, one of the really interesting studies was of dogs. Dogs who run on lawns that were treated with 2,4-D. And first of all, you can find it in their urine, as a study found, and these dogs had an excess of lymphoma, like humans, in a sense, and we're pretty sure the dogs weren't applying. I think that's what you're asking, those sort of studies, right?
But also for people who are, who've had Chemlon, people who've had Chemlon for like 10 years.
I personally don't know of any studies like that. Doesn't mean there aren't out there, but it doesn't ring a bell to me. There are, in our study of agriculture health study of farmers and their spouses, we're now just getting ready where we can look at bystander exposure to the spouses if they weren't applying themselves. Sometimes spouses do, and so they're an applicator also. But many times the spouses don't, and the children by trying to get at the different levels that might come in from different bystander exposures, some drift, some track home, and that sort of thing. And we have people working on developing a exposure algorithm so we can look at that end of, in this prospective cohort with a wide variety of outcomes. Yes, here.
So can you tell me what the role of the studies looking at endocrine disruption is having in this kind of, endocrine disruption, because you've been talking about prostate and thyroid and breast and ovarian. Many of these are endocrine, based, and I'm curious what the role is.
Well, I think the sort of the underlying assumption, mechanistic model that you follow, that if that is occurring, it's through the endocrine pathway that it operates in some way. And there's evidence, some evidence that that is the case from mechanistic studies and animal studies. But in human studies, it's not good for cancer. It's probably better for other things, which actually, in all honesty, I know less about. You may have other speakers who know more about that. But the model, I think, seems sound. Any things that will interfere with the normal endocrine processes in our body is hard to believe that's a good thing. So whether or not it's big enough to do anything, I don't know, but that's the sort of information to be useful because there's plenty of pesticides and other chemicals, I might add, that have these effects.
So in an IARC working group, are you looking more at endocrine disruption studies than you used to?
Well, IARC for cancer, in one of these IARC working groups, you look at everything. There's nothing that's left out. Yeah, so it looks at everything. It wasn't a big contributor to the decision for the IARC group that I was on, or the second one in that year either, because there wasn't much information. It doesn't mean it might not fit in later on, but most of the studies didn't do that. And many of the epi studies couldn't do it at all. All you knew is people were exposed, and you looked at outcome. You didn't know anything about what went on in between. I mean, that's okay. There's nothing wrong with that. It's just every study can't do everything. Yeah. Yes, right back here.
Is it too soon to start having data on applicators of glyphosate for the cancers?
No, that would be, are you asking us like from a research point of view?
Yeah.
Yeah, absolutely, that'd be great. Absolutely, yeah. It's, I mean, that's what I did. My whole career was research. I'm always up for doing more research, but that's a group where it would be good to look at. Now, out of the agricultural health study, these are applicators. They don't apply just glyphosate, but they apply other things. And it's worthwhile you look for groups like that. The better you can target and narrow the exposures, the easier it is to understand what's happened, and actually the easier it is to make the argument of what you ought to do. I'm not sure it's not the way humans are exposed, but in the thinking of society, how you deal with things, you like to see single exposures. Just let me give an example of this. So everybody believes, I'm sure, that smoking causes lung cancer. And so do you think that that's a problem in interpreting those data because humans also have other exposures? They get exposed to bacteria, they have some pesticide exposure, they drink water with chlorinated products in. I'm sure none of you think this is a problem. It's because so much evidence has been gathered, the risks are so big that it just blows the rest of it away. But again, I keep saying it, there are no humans with individual exposures.
But you haven't started collecting the data on the number of times, say, your GMO farmers are using the glyphosate.
I don't actually know of studies looking at just the genetic modified angle. There may be, but I'm not familiar with that for cancer. And for what we do, many of the people who applied glyphosate in Iowa and North Carolina were doing it on genetic modified soybeans first, but they may have had exposure to glyphosate before that. We started. So I don't know of any cancer information that deals just with the genetic modification crops and the herbicides that go with them, although that's certainly doable now because it's so widely done. Back here.
I've seen literature criticizing some glyphosate studies as dealing only with the isolated glyphosate and not such things as surfactants and other so-called inert ingredients in the pesticides as they're formulated for sale. Could you comment on whether you consider that to be a significant issue with the glyphosate studies, or and/or studies across the board of other pesticides?
Pesticides have this problem. I don't know of any single pesticide in which only the active ingredient is applied. You know, there's always other stuff in it, I think. And so, yeah, it is a problem. But that's actually, then when you couple bioassays and experimental studies together, they can control this. Human studies you can't. You must take what people do, which means they go somewhere, they buy a jug of something or other, and they apply it either individually on their lawn or on their farm, and whatever's in there's in there. You know, now, some things do change. Some different pesticides have different sort of forms of the active ingredient. Actually, glyphosate's one. I mean, there's been more than one form. 2,4-D's that way. There's an acid and there's an ester. And so there are ways to get at it even in human studies, but the real way that helps is to look at the experimental end of things and see if it's only in the sort of the sellable product or if it's in the active ingredient. With glyphosate, it was both for experimental studies. I saw something else, and I think maybe back there.
You know, we've been looking at pesticides for the last 35 years, first in the Sustainable Cotton Project and then in our project in Vermont called Vermont Regeneration Vermont. And the big problem we have is bad data. The USDA National Agricultural Statistics data we evaluated from 1995 to 2004, and we found out, because California has pesticide use reports, that the USDA data only accounted for 53% of use over that 10-year period. And in 1 year, it was only 38%. And we found the same thing in Vermont. And only a few states in the United States even collect pesticide use data, and most of the data is based on surveys that the USDA does. And is the cancer data, my question is the cancer data as bad as the pesticide use data?
Well, I think it depends on, you have to go sort of study by study. It's the right question. It's the critical thing. I actually have a few slides I think I may use at one of the workshops about the impact of what misclassification does. It's horrendous. I mean, it's so awful that I'm surprised sometimes we find anything. So yes, that is a problem. But these studies that are done for cancer, it's not taking aggregate data from survey that was done. We ask the people directly or gather information directly on the individual person. Sometimes that works. It probably wouldn't work if you were trying to study cancer in farm workers. But it does work for owner-operators. Owner-operators, they know what they did. I mean, they really do know what they did. This is important. I mean, it costs a lot of money. If they get it wrong, they really screw things up and they go broke. So these people can tell you what they use. In fact, almost everyone can tell you how much they used on the South 40 last year. Now, as time passes, they forget, just the way all of us do. But if you do the study in that way, the initial things is very good and much better than the survey things. But there's always exposure misclassification, so that's a good question. Yeah, right there.
Any studies of condominium unit owners? In other words, someone who's here in the audience lives in a condominium association that 4 times a year likes to apply a mixture of 3 different herbicides and insecticides and stuff because there might be a living bug in the grass, and so that's part of the reason she's here.
I don't know of any specifically with that group, but there have been studies of people sort of not applicators, not farmers, not groups that you think are using this for the way they earn their living. And generally, for cancer in general, those studies don't show anything. Now, there's a lot of things that might explain that. One, the amount of exposure misclassification is like to be an order of magnitude greater than farmers that I just talked about. And secondly, exposures are on average a lot less. And so it got 2 things going there which might drive it towards a null even if it was real.